All this is Dr. Mobeen Sayed from Drbeen.com. Welcome to one more show. Today we have a stellar guest with us, and I think somebody who actually has a promise, whose company has a promise to lead us out of the situation that we are all in. So we have Dr. Herrera with us. Let me very quickly introduce Dr. Herrera's, give you his introduction. So from his website, so let me show you the website as well, and all of these links are present in the description. So here, as a PhD student, Herrera developed T-cell based diagnostic tool. So many of the cool beans here, we know about the T cells. So T-cell based diagnostic tools for Zika, dengue, and Ebola viruses that were implemented in local hospital and labs. Now he's the co-founder and CEO of E25Bio Inc, which aims to develop affordable, rapid diagnostic tests for patients. In 2019, he was named Forbes 30 under 30 healthcare. And check this out, check out his education. He earned a Bachelor of Science, New Mexico State University, Master of Theological Studies, Harvard Divinity School, PhD in Biological Sciences in Public Health, Immunology, and Infectious Diseases, Harvard University. What a star we have today with us. So welcome, Dr. Herrera. Can you tell us a little bit about yourself and your company?
Sure. Well, great. Thank you for having me on your channel. Happy to be here to discuss about, most importantly, E25Bio and the work that we're doing. A little bit about myself. Originally, as you had mentioned, originally from New Mexico, but moved to Boston for school. I studied infectious disease immunology at Harvard University, and particularly in Phyllis Conkey lab at Harvard T.H. Chan School of Public Health, where I was really interested in studying T-cell responses to emerging infectious pathogens. In 2018, along with Professor Lee Gerke from Harvard and MIT and Dr. Irene Bosch, we spun the company out of the Professor Gerke's lab in 2018 to develop rapid diagnostics for epidemic fever viruses. Prior to COVID, we were very interested in, and still are interested in, developing rapid diagnostic tests for mosquito-transmitted viruses such as dengue viruses, Zika, and chikungunya virus. These are mosquito-transmitted viruses. In the case of dengue virus, causes 400 million infections worldwide each year. So it's a significant global health burden. And then moreover, in response to the COVID-19 pandemic, we sought to develop a rapid diagnostic test for COVID-19. And I guess we'll talk a little bit about that today.
Absolutely. So very quickly, from the Dr. Bean side, so we are a tribe that we call ourselves Cool Beans. We are about 1 million in strength over Facebook and YouTube and drbeen.com site. We have been looking forward to having you with us for a long time. The reason for that is, in the COVID-19's context, the reason for that is that we are looking to figure out, is there some way that we can at home know if we are sick or not, if we have COVID-19 or not? And then hopefully once we know it, then we can figure out how to move forward and how to aggressively manage the situation. So I think that while the vaccines are being developed and while there are recipes and protocols being developed, testing and knowledge of we being sick or not is the paramount thing. So I believe that the work that you are doing is going to be the leading work for all of us to get out of this situation. So with this, let's start our discussion. I'm going to very quickly just show your site around for a second. So, excuse me. So we have some smoke here in California, and that irritates my throat. So my apologies in advance for occasionally coughing. So here is Cool Bean. So here is the E25Bio's Twitter feed. So please, I would request you to follow him. Then here is, as you can see, Dr. Herrera 29. So this is Forbes 30 under 30 for 2019. So here he is. This is about the E25Bio, and the description for all of these links, the links to all of these are in the description for this video. This is Dr. Herrera's about him. And then finally, here are the products that we are, especially the SARS-CoV-2 product that we're going to talk about today. So with this, let's go back to Dr. Herrera. So Dr. Herrera, tell us this. Is this something that has been functional? Have you tried it, tested? What is it? What is this kit? Why is it going to work in 5 to 15 minutes? Why do you not need any machinery? What is it?
That's right. So E25Bio, like all of our other tests, it's to enable public health response in a variety of ways. And really what E25Bio, the reason why we created E25Bio was to provide affordable rapid diagnostics for all individuals. That includes people at the top, such as many individuals living in the United States and elsewhere, as well as many other individuals in resource-constrained regions, especially in resource-constrained regions. And so what E25Bio has done is we've taken the modern centralized laboratory and we've shrunk it into a paper-based strip. And so you see that here. I show it, and I've showed it a couple of times in a couple of videos, as well as you can see that in various of our news articles online. But ultimately, what this paper-based strip is a laboratory on a strip. And ultimately, what this allows us to do is to identify active infections in real time. And I think that's the operative that is the operative word, active infections in real time. So while PCR, polymerase chain reaction, is really great at identifying, it's a super high sensitive, highly specific test, great at also identifying active infections. It requires a laboratory and trained personnel to perform this test. Antibody-based tests, also really great. They tell an individual that they were infected, they previously were infected, they may be protected for a certain number of months, but they do not tell you that you are infected. And what E25Bio has done is we've shrunk, again, a laboratory into a simple paper-based strip, which will allow an individual to know whether they are infected or not. And because of the simplicity of this test, it does not require a reader. One is able to visually realize whether they are infected or not with the presence of a single band indicating no infection or double band indicating yes, you are infected. What that will allow us to do is then to bring this technology into our homes and to really do these things at high frequency, because this is what it is all about. We are changing what E25Bio is postulating, and what we would like to advocate for is for a shift in the way that we think about doing infectious disease diagnosis in the United States, from it currently being limited to and centralized to the laboratory. And rather, I think COVID has demonstrated that we need to think about things a little differently. And so rather, we want to take these technologies into the home. They are robust. They're sensitive and specific enough to take into the home, and that only gets better when you perform the test multiple times per week, for example. And so this type of testing technology allows that, and that's why I think we're onto something interesting here.
Got it. So, Dr. Herrera, I was trying to figure out how to zoom you in onto the video, and I could not. And at that time, I just started playing with it. So, can you show us that strip once more?
Yeah, sure.
Excellent. And so tell me this, and there are a few comments here as well from the Cool Beans as well. The first question is, there are a few comments that say that this would only work if there is a larger viral load. So, excuse me. So in terms of sensitivity and specificity, how does it compare to the PCR tests?
Sure. So I think one thing that's very important to realize is that in general, antigen-based tests, so again, the type of technology that E25Bio produces, as well as others, in general, antigen-based tests will never be as sensitive or specific as your gold standard method, which is known as polymerase chain reaction. Polymerase chain reaction is a method in which you're detecting viral nucleic acids and amplifying them. So you're turning two viral copies, genome copies or gene copies, to 4, 4 to 8, 8 to 16, 16 to 32, and so on. And over the course of this experiment, it's an amplification method, which is why this method can be so sensitive and specific. Rather, an antigen-based test, that is not an amplifying method. And rather, using one of these types of testing technologies, we are able to detect antigen that is present at any given time during the course of infection, and particularly during the active phase of infection. So we're talking in terms of a viral infection between the first 1 to 7 days of infection. That's when I think we can really capture enough viral antigen to let an individual know whether they are infected or not using one of these paper-based strips.
Got it.
So then the question is, how sensitive are these tests? And what I will say over the course of years of validating these tests on human samples is that, again, in general, they will never be as sensitive or specific as gold standard PCR. However, this is the caveat. When performed during that active phase of infection, when the patient is harboring the largest quantities of virus, or during that early symptomatic phase, so again, 1 to 7 days of infection, when a patient is experiencing fever, cough, etc., our tests can be as specific or more specific than the clinical gold standard, which is PCR. This is results that we've obtained over our own clinical studies, and some of the tests that have been that have obtained emergency use authorization by the FDA. They have also shown that these tests can be more sensitive than PCR. And so I think the one important aspect here is, again, rethinking, reshifting, changing the paradigm in which, or of which, how we think about doing diagnosis in the United States, taking it from the laboratory and into the homes. And when you take it into the homes, it's really about that high frequency, catching people during the symptomatic phase, not post-symptomatic phase, such that you can get the best results on one of these tests.
I love it. And for the cool beans over here, so we have been talking about asymptomatic stage, and then the symptoms develop, and then post-symptom where there is immune dysregulation. And we always have been talking about this, that the disease needs to be caught early on. It needs to be aggressively treated early on. So if I am, let's say I get symptoms and I have no idea, is it cold, is it rhinovirus, is it influenza, what is it, or is it COVID? I think at that time, if I have this kit available and I can very quickly verify that this may be COVID-19, then I can aggressively start taking the next steps to make sure that I do not end up in a dysregulated state. So I think this is a crucial test for us. I have a couple of more questions here. One of the questions I saw in the stream here, and you kind of touched on that as well. Is it necessary that we have to be in the active phase, number one? And number two, does it only identify the active live virus, or is it possible that those remnants of the viruses after some months that are still present, it would pick those up as well? What kind? So can you please comment on this?
Those are really great questions, and I will hearken it back to the current testing schema which we're currently experiencing, which is in the United States and most countries around the world. The way that we are performing diagnosis for COVID is only during the symptomatic phase. So the moment an individual is experiencing fever, cough, and there's reasonable measures to say that this individual is sick, they will then report to the hospital, to the doctor, to be screened for diagnosis. Ultimately, what we have seen over the course of several studies is that patients are going to the doctor and actually they're being given the clinical gold standard, which is PCR, too late. So post 7 post 7 days of fever infection. And ultimately, what that means is we have caught the infection too late, meaning for the first 7 days, yes, this individual was sick. They reported to the doctor. And yes, PCR is so sensitive that you can capture the infection after the 7 days. However, what ends up occurring with COVID-19 is that post 7 days, post 10 days, this patient is no longer infectious. So then what good is it to tell a patient that they were infected and they're no longer infectious? So then again, it's shifting the paradigm in the way that we do this type of screening. And I think what is so important with COVID, what we've learned over the almost 9 months now that we have been learning from this virus and therefore counteracting against this virus, is that we need to catch this virus early enough. And that starts from the pre-symptomatic, asymptomatic phases especially, and also during the first 7 days of infection. And what I will say is that, again, antigen-based technology, antigen-based tests, such as the one that E25Bio has created, these tests are very, very good, as sensitive, if not better than PCR, at detecting active infections compared to the clinical gold standard. And what that will allow us to do when these tests are being performed at high enough frequency such that individuals are identifying themselves early during the course of infection, then what that allows us to do is isolate them. And we can isolate them within enough time such that they're not infecting others around them. And so I think that's really what we hope to achieve with this type of testing technology. It's not to replace PCR. It's not to replace antibody-based tests. Again, all of these tests are very important in their given settings. However, what we are advocating for is that these tests can be used at high enough frequency, and the only way to do that is that when they're distributed in mass, and particularly when patients are taking this technology into their own hands, performing self-testing at home. That's really one way to combat this virus, to reduce infections. And when you reduce infections, of course, you also reduce hospitalizations, and then ultimately
Absolutely. So for the cool beans, this I'm going to repeat this. This is our ticket towards saving our lives and the ones near us, our loved ones, and the humanity in general. Catch it early, find it early, and then treat it early. Dr. Herrera, we are actually believers of aggressively treating early as well, instead of knowing that I have COVID-19 and just waiting. So waiting and isolating is great because we're not going to give it to others, but we would have a chance to treat ourselves early as well. So this is the ticket. And now, couple of more questions now. One question that I want to ask you, this is something I wanted to ask in the beginning as well. When can we place the orders for this test? How expensive is this test? When can we get it? I think that is the most important thing.
Sure. No, those are obviously very important. Those are, if not the most important questions. So ultimately, E25Bio has to work here in the United States. We're obligated to work with our regulatory agencies, and that is the Food and Drug Administration. The FDA has set a number of policies which guides antigen-based manufacturers towards being able to approve their test for commercial sale. And because of that, there has been ongoing policy changes in order for one to get these products into the market. And so what I will say is, to date, there's about 4 to 5 different companies that have gotten their tests to the market. A majority of those tests, however, are confined to laboratories or CLIA waive settings because they require a reader. There is one company that has been able to get their product to the market which does not require a reader. However, that test is not being sold for at-home use. And so what I'm trying to say is that E25Bio, currently, we are working very hard on trying to get our products to the market. Actually, FDA is, we're working with the FDA on a number of our products. And then ultimately, we hope to get this product into the homes, although no company has yet been able to get a product for COVID into the homes. I'm very optimistic about when this timeline could occur, not only for my company, but for other companies as well. At the end of the day, I'm a public health practitioner. I think as many tests that can come to market is going to save many lives in this country and other countries around us. So I'm advocating for as many tests to come to market. But ultimately, with regards to the E25Bio test, I hope that our test gets to market very soon, within the next few weeks to the next month. And then ultimately, I think it's going to take some time for these companies to get into homes. I'm very optimistic that it will occur by the end of the year, if not early next year. But I think, as we all realize, I think COVID is going to be an ongoing issue for the United States as well as the world. And so these will be much needed now and into the future, especially despite a vaccine coming to the market. Now, with regards to price, E25Bio, since our launch in 2018, again, not for COVID, but for the mosquito transmitted viral line, dengue, Zika, chikungunya, we have always been socially oriented, meaning we want to bring affordable rapid diagnostic tests to the market for all people. And because of that, we aim to price point these diagnostics such that all people would be able to access them. And likely these are going to be in the single-digit dollar amounts, US dollar amounts. However, of course, we are going to be competitive on the market. And so I think, as you know, the Abbott price point has come out, and we will be in line with others.
Makes sense. And once again, I understand that the approvals are needed. Only then you can come out and sell it out to us for at home. I still believe that if we have to sponsor companies, if we have to give funds to the companies, that, for example, for vaccine companies, we're giving billions of dollars. These are the companies like E25Bio that need to be supported at this time, so they can rapidly develop kits that can be present with us, maybe 100 kits per home, maybe $100, $200, so that anytime we suspect that I may have it, I have cold symptoms, I can very quickly go and check myself and at least understand, maybe if I have it, even if the sensitivity may be lesser than PCR, if I suspect that I have it, then I can take appropriate actions instead of just sitting at home and thinking it may be flu, especially now that the flu season is upon us. So this is a great thing that you're doing. I have a question here from James Kelly, and I think you've just talked about it. James said that, so assuming one of these was available, can this be simply used at home and diagnosed with simply reading a single or double band? So James, yes, Dr. Herrera explained that it is single or double band, or does it need to be analyzed in a lab? So no analysis, right? Question 2, what are the manufacturing challenges and expected possible dates? So how about that?
Yes,
the manufacturing challenge.
That's actually, I think that's one very important point that is missing from a lot of the conversations that have been had, which is, it's easy to say that, we need to do high frequency testing in homes. It's a mission that we can work towards, but what exactly does that mean, and how exactly do we get there? In the United States, there are roughly 320 million people inhabitants of the United States. So how do we produce 320 million tests per month, for example? That's a significant amount of tests that would need to be not only manufactured, but then also distributed. And so I think that's an important conversation that we should be having. And what I will say is that the underlying reagents that goes into these tests, the underlying reagents is not what creates the bottleneck. But where the bottleneck lies is on the manufacturing as well as the distribution. And so just like the United States is investing millions, hundreds of millions, and billions of dollars in vaccine development, I think similarly, the United States and other foreign governments should be investing in testing capacity, particularly for manufacturing and particularly for distribution. And so, this is just to be provocative, but why doesn't every state in the United States have a manufacturing facility such that they can manufacture their own tests and then distribute widely in their state, right? Why are we manufacturing in Massachusetts and then ultimately would have to ship to my hometown, which is where I was born and raised in New Mexico? Rather, why can't New Mexico have their own manufacturing facility? And so I think, again, it's about being strategic in the way that we're going to manufacture these things and then also distribute them. And I think really that's the conversation that's missing. And ultimately what it is going to rely on, it's going to rely on governmental support, whether that is monetary support to produce more manufacturing facilities for these types of tests as well as others, and then being able to distribute these. And what I will say is that every test that we produce for COVID, it takes a test away from a different disease indication, such as diabetes, cancer, etc. So it's important that we improve our overall manufacturing and distribution capacity and capabilities, because COVID is not going to be the only infectious disease pandemic that we have to deal with. There will be more in the future, as evidenced by the recent outbreaks in the past 5 years or 7 years, Zika, Ebola, now COVID. It's important that we really advocate for improved public health preparedness in the United States, of course, but also in other countries.
Got it. Couple of questions then. One question which is really very interesting. It is from M-Ball here. What is the shelf life of this? And then another one, that if you come up negative, do you need to do the test again? Yeah. And I would suspect that if you have so many tests at home and if it is cheap, then you can probably continue to do more. So that's right. What do you think?
Yeah, those are great questions. So I'll start with the first question. Lateral flow tests. So this type of technology, it's a pretty old technology. It started with the pregnancy tests, and obviously I think many of us in one shape or form are familiar with the pregnancy test, and these tests can be purchased over the counter, right? You can purchase at a local pharmacy store, and so the ultimate goal is to bring these to the market in that way. The typical shelf life for these types of tests is between 1 and 2 years, which again, that's a pretty good shelf life.
Absolutely.
And then ultimately, what ends up occurring when you are negative? And I think the point here is that because COVID and because many of these viral diseases are so dynamic, the point is one can increase sensitivity or accuracy of a test by performing the test more frequently. E25Bio recently archived a publication at medRxiv. It's a preprint currently where we're showing different sensitivity levels and how sensitivity fluctuates depending on frequency. And what I will say is that, again, the frequency is what matters more so than test accuracy, particularly for antigen-based tests. So if you're negative on a single day, if there's reason to believe that you have been exposed to an individual, or say you went shopping because you need to shop for groceries, that is a high-risk area. So why not test for the next few days? And by being able to bring these testing technologies into the home, I think that would be one way to improve overall accuracy for the test, but then moreover, improving overall public health preparedness.
Got it. So this test, which is with you right now, is this a functional test? Can you actually right now use it?
That's right.
Is it saliva based?
I just actually ran the test. So previously I was holding up the paper-based strip alone. Now I put the test into some liquid, and then ultimately it ran, and you can see a single band there. Of course,
Yes.
it's glare, but there's one single band which indicates a negative result. Had a person been positive, then you would see two bands similar to that band. So
And is this your saliva-based test? So did you touch it on your tongue?
no, so this is not a saliva-based test, but rather it's a nasal test.
I
So it's
see.
talking about
Hmm.
anterior nares. It
Hmm.
does not
You
require
touch it here.
the uncomfortable nasal
Hmm.
pharyngeal swab, but
Hmm.
rather would
Hmm.
be a simple nasal swab. So we're
Oh,
talking
wow.
about just right in the anterior nares compartment.
Oh, wow. So you are the smarty pants who has the test with you right now, and you can actually test it every day if you want it. This is excellent. So I totally suspect that once your tests are at a stage where you can start selling them, people just like in front of Apple stores, they camp out to get the test. Probably will be camping out in front of your company saying we want the tests as well. This is excellent. Tell me this: are you getting enough funds and support to ramp up your production and your processes? How are you? Uh, I'm seeing again in case of vaccinations or vaccines, we are throwing millions and billions of dollars. Are you being supported?
E25Bio has been fortunate enough to be supported by some really great investors.
Hmm.
Whether that is institutional investors from venture capital or government investors from the NIH or the Gates Foundation, we're very grateful for the support that these institutions have provided to the company. But what I will say is it is not enough. As I had mentioned before, we are throwing billions of dollars into vaccine development, and I think that is rightly done. We need to be investing billions of dollars in improving vaccines, not only for COVID, but for the emerging virus that is going to emerge next year and in 5 years and in 10 years. In addition to those funds, I also think that we need to invest more dollars into improving our diagnostic capabilities, not only for E25Bio, but for other companies as well, and for other academic labs who study a lot of these that help companies like mine improve our diagnostic technology. So there is not, in summary and in short, I don't think enough money is being poured into diagnosis like there is into vaccines. And what I will say is that simple tests like this can actually save lives when done at frequency, at high frequency. We just need to create awareness for this types of testing technology, but then moreover, um, uh, invest dollars such that one, we can increase manufacturing, improve our distribution strategies, et cetera, et cetera. So yes, I think that more money needs to go into diagnostic testing technologies in particular. I think you're on mute.
Thank you very much. Thank you. So for the cool beans here, remember once we talked about Dr. Jacobs' company, which was making polyclonal antibodies, and I was saying that we need to raise funds for them. This is another company, E25Bio, that if we think about it strategically, that how do we get out of this mess? Then knowing quickly, rapidly at home that if we are sick or not with COVID-19 is paramount for us. And for that, here is a company that can do this, that can give us things, the tools that can help us stay safe and protected right in the beginning. And so we should be able to do whatever we can. I don't know if we should do some GoFundMe type thing or if we should go and influence the others, but this is the company to support to get us further forward. Thank you very much for your work, Dr. Herrera. Tell me this. There is a question here, and I think you've touched on this before as well. So Jude says that COVID-19, how many days after the symptoms someone tested, 5 days, 7 days, what time will it give optimum results? And I think you talked about it before too.
Ultimately, that's a really great question. And what I will say, again, our current testing schema in the United States as well as most other countries outside of the United States, the way that we are doing diagnosis is symptom based. So the moment a person presents with symptoms and um, is believes that they have COVID, then they report to the hospital or to the doctor or to the clinic such that they can get the gold standard, which is PCR. However, what we have seen in many studies is that time point, it's too late. This person is outside of the active phase of infection and they are no longer infectious. So for those first 7 days, they infected all of their family members. And ultimately, what you don't get is a reduction of that curve. You get a continued exponential growth, which we have seen for quite a long period of time here in the United States. What an antigen-based test will allow is high-frequency testing, particularly during the active phase of infection. An active phase, what we have seen with COVID, can occur during an asymptomatic presentation. So you can be exposed to the virus. Within a couple of days, you're either presenting with symptoms or not. And however, you have a very limited window in which one is able to detect infection or not. But if you are infected, there's a limited window, particularly 7 days post symptoms onset. But again, a lot of individuals don't ever even experience symptoms, which is why we're in this terrible situation that we find ourselves in, because COVID is such a tricky disease to handle. But again, typically post symptoms onset with an antigen-based test, you are able to detect with high enough sensitivity that it's meaningful for 7 days. After 7 days, sensitivity and accuracy will be too low, and therefore you are going to require PCR. But again, it's debatable. However, post 10 days of symptoms onset, patients are thought to not be infectious any longer. So then what good is it for a patient to go to a hospital, obtain PCR, which by the way is quite expensive and laboratory intensive, to be told that they are infected, but again, they're no longer infectious. So then, again, it's important to identify infections during the active phase such that we can institute some measures to control active spread of the virus. And then ultimately, when you control spread, you're reducing deaths. And that's the ultimate goal here.
Absolutely. And I would just add the same point I had done before. We here in Cool Beans, in our tribe, we believe that right in the early stages, we have to aggressively manage with ivermectin or hydroxychloroquine or vitamin Ds or whatever else. We have to take care of it early. But for that, we need to understand that are we ill with it? And so this test actually allows us to know that and then manage it aggressively. So a couple of questions now. One question from a Cool Bean and then one question from me. I'm just going to try to nerd out with you on the medicine side. So one question from Chantel here. Is there anything that would affect the result of this test, such as cold, flu, measles, chickenpox, and other conditions? Is there any cross-reactivity?
Yeah, that's a great question. And part of getting these tests approved for sale in the United States, at least, we have to take these tests through very rigorous analytical performance studies. And as part of those analytical performance studies, we have to ensure that these tests do not cross-react with many other microorganisms, both in viral and bacterial in nature. And so our test has been shown to be very specific for SARS coronaviruses. It does not cross-react with many other respiratory viruses, such as influenza or other bacterial infections. So in short, no, these tests do not cross-react with the chickenpox, measles, etc. We have taken them through pretty rigorous studies to show that they do not. And then, of course, it's something that the FDA is very keen on evaluating prior to approval of any test.
Got it. So one last question now, and thank you very much. Before we finish, thank you very much for your time. The last question is this. So how does this test work? So we know that in PCR, we amplify the RNA and then we try to figure it out. What is your reagent made of? What does it do to the virus? Does it bind to the spike protein or to the envelope? What is the, zoom us into the functional part of this thing, this test?
Sure. I'm sure that all of the cool beans will be very excited about this answer. So I had mentioned that this test has taken a modern, centralized, sophisticated laboratory, and we've shrunk it onto a paper-based strip. And that's ultimately what we've done. It's a paper-based strip that contains some very sophisticated molecules and components that then allows us to identify infections. And so what those molecules and components are called monoclonal antibodies. These monoclonal antibodies, they are like hooks at the end of a fishing rod. And however, these monoclonal antibodies, these hooks are very specific for any given target. So rather than when you go fishing, you don't know if you're going to catch a trout or a salmon. Here, we know we've engineered the reagents such that it will always catch a salmon, and that's it. And in this case, we're very interested. That's a poor analogy, so forgive me for bringing that analogy to the table. But however, what I'm saying is that we have developed, we have isolated, and we're using very specific monoclonal antibodies. These molecules are very specific for SARS-CoV spike antigen. On the surface of the virus, think of its skin as the surface. It has these proteins called spike glycoproteins, and it is what covers its shell. And so our monoclonal antibodies are very specific for that spike protein. And so, for example, if I am experiencing COVID, or if a person is experiencing COVID, the virus is secreting spike into the nasopharyngeal compartment. So you capture those spike proteins with the nasal swab, you dip it into this tube, and then you insert the dipstick as you see inside this tube. And ultimately, what will happen is that the fluid which came off of your nose, which contains those spike proteins, will pass through the paper-based strip and ultimately will interact with those molecules known as the monoclonal antibodies. And when the monoclonal antibodies interact with the spike protein, it'll capture them like a sandwich, two breads, and then the meat in between the sandwich is the spike protein. It'll capture that, a pH change signals, which then creates the presence of that single band or a double band. And if we see that double band, then ultimately the test is identifying an individual who is experiencing active COVID infection. And so therefore, we would then isolate those individuals, quarantine those individuals such that they don't spread the virus to others around them.
I love it. I love it. And what a genius you and your company is that has come up with this. I had promised this was my last question, but I see a couple of questions that are really important, and they are connected with each other as well. And I think only you can answer them. One of them is, we have human coronaviruses as well. And the other one is that we have SARS-CoV-2, which is mutating as well. So will this test kit also detect human coronavirus, and would that become the false positive? And what would happen when the virus is changing and mutating?
Sure. So, well, SARS-CoV-2, which causes COVID-19, right? That is a human coronavirus. It's a coronavirus. SARS-CoV-2 is a human coronavirus that causes human disease. So ultimately, again, we've engineered this test to detect SARS coronavirus, the human coronavirus, the SARS-CoV-2, which causes COVID-19. However, this test does not cross-react with other coronaviruses, ones particularly that cause other human disease, such as MERS. So again, it's very specific for SARS coronaviruses. However, it doesn't cross-react with other human coronaviruses, such as 229E, OC43. These are other coronavirus strains that can cause disease in humans. However, this test does not cross-react with them. So it's very specific for SARS coronavirus.
Very good. So thank you very much. And one more question, and then we stop. Sure. Carolyn says, how can we support Dr. Herrera's work? What can we do?
Sure. Well, I think the most simple form is to call your senators, call your local government, demand that these tests get approved for not only over-the-counter use, but also for ultimately at-home use. What we have shown, what our models have shown, what our clinical studies have shown is that these tests, when performed at high enough frequency, are as good, if not better, than clinical gold standard polymerase chain reaction at detecting COVID-19 infections. Ultimately, when you detect infections, you reduce hospitalizations, and ultimately you reduce deaths in the United States. We have experienced too many people who have succumbed to disease, and ultimately these simple technologies can save lives. And I think that is what we should be advocating for, protesting for with our local governments, such that they allow these tests to come into the home once, of course, they pass some reasonable regulatory standard, which is a high enough sensitivity, high enough specificity. I'm not asking for us to bypass those standards and those metrics, but rather once we have, it's going to be important for us to get these into the homes. And I think if we have enough people advocating for this, we will be able to get there. And I'm optimistic that we will very soon.
Excellent. So Dr. Herrera, once again, thank you very much. I know that you're on the East Coast. It is now going to be about 10 o'clock on your side. So thank you for your time. Thank you for coming and having this discussion with the Cool Beans. And we are all going to be waiting for this test to become approved and to use it. Thank you very much.
Thank you so much. And thanks for everybody for tuning in.